Hi, and thank you everybody for attending here today and tuning in. My name is Nick Pervolarakis. I'm a computational biologist here at Active Motif, and today I'm going to be discussing the topic of interacting with data on the UCSC Genome Browser and what that can do for you and what that means. And so a brief overview of our agenda today is we'll be walking through, firstly, a little bit of details about the browser and the data that we'll be working with. How to actually access the genome browser, walk through example of uploading data, as well as specific inquiries that you can use this tool for, and how to go about that. And we'll bring it on home with a summary at the end. And so, without further ado, what is the UCSC Genome Browser? It is first and foremost a powerful tool for data visualization. So it's something that is very widely used in the field of epigenetics and beyond to create publication quality figures and visualize a lot of signal differences for specific genes or loci on the genome for data like ATAC, ChIP-seq, cut and tag, things that are largely peak-based assays and the like. And so what do I mean by this, or what data is sort of amenable to being viewed on the browser? I think today we're going to be using ATAC specifically, but the same holds true for ChIP-seq or those other assays that I had also mentioned. And so how we get data that's usable on the genome browser is first we start with what are called FASTQ files. So these are your sort of raw unaligned reads that have been demultiplexed after sequencing. We take those and align them to a reference genome, resulting in a BAM file. So now we have the read information and where on the genome the physically aligns to. And the next step is what's called peak calling, where we take these aligned reads and we then, using a software called Max2, primarily look for regions of significant signal enrichment over background within that sample. And so those regions where we have more reads that aligned as compared to others are what are then called peaks, which become the unit of downstream analyses and tertiary questions that you're able to ask. And so that information is ultimately converted into a file format called a BigWig file that is binarized and is a very small and sort of lightweight format that allows for ease of visualization and quick turnaround time for queries into what would otherwise be a very large dense data file that is your BAM file. And so that's what we'll be working with today for an example ATAC data set that we've generated in-house. And so the first step of actually using the UCSC Genome Browser is to travel to this URL, the genome.ucsc.edu, and you will land on a page that looks a lot like this. There's a lot of information here. You're not going to be, for today's example, not going to be interacting with any of these others besides this Genome Browser button. And so if you press this, as we've highlighted in red here, it will take you to a page that looks a lot like this. And so I don't have any other previous data loaded in here, and this is just sort of a blank, empty view on what the browser can look like and some options that you can interact with for it. There's a lot of buttons here, but the one that you're going to be most interested in to actually start interacting with your data is this Add Custom Tracks, where upon pressing this will take you to another page that will let you successfully load in the data that you're interested in viewing. And so that page looks a lot like this, depending on your browser settings and things like that. Any internet browser will do. Internet Explorer, Edge, Chrome, Firefox, so on and so forth, Safari. And so they'll all be very similar. And the most important thing to start when we're on this page is firstly selecting the genome that your data was aligned to properly. And so I mentioned earlier that that's an integral step of any analysis. And so you have to ensure that the genome that you're pulling up to view your data on matches the genome that it was aligned to in the first place, because there can be conflicts with chromosome size, or if it's across different species, like obviously that's going to cause some problems. And so check these boxes and make sure that this is the correct information. Here we're using the human genome build, GRCH38, for this data analysis. And so next, we're actually going to be uploading the correct files to perform our actual viewing of the data. And so that is done here in this choose file portion. So there's a handy little button that you press, and it pulls up a window to select your file of interest. And so what I have here is an example delivery of what Active Motif bioinformatic analysis actually looks like. And so there's a lot of files. You're not going to be using every single one of them. We'll save it for another time to maybe walk through other specifics here. But today we're going to be interested in one particular file that I've highlighted here that's entitled AM sample data ATAC. And the important part is UCSC tracks_bw.bed. And so this file contains your track information, and it also importantly points to your BigWig files, like I've mentioned, that we are currently hosting on our FTP site that we deliver, a completed analysis to for researchers' projects. And so that's the important part for viewing anything here is this hosting on a remote site to make ease of use for access to these files. And so we've taken care of that hosting for you, and you can select this file and go from there. And so after you click the right one and you press open, it will load it into the box, and you go ahead and press submit and begin the process of visualization. And so when you press submit, you're going to see a page that looks like this. And it's not going to look exactly the same. The basic structure will be there, but I'm going to sort of highlight where things might be a little bit different. So for our analyses, there are these, in this example data set, 4 different samples that were analyzed. And so firstly, we have what's called the merged regions track. So basically for all of the peaks that were called across all of the samples, this will have some information for that on their position. And then now we go into pairs of files that correspond to, or pairs of tracks, I should say, that correspond to each sample analyzed. And so we have the BigWig information, and then we also have information regarding the position of actual peaks in the data set. And so this pair is repeated for every sample. And so it's starting on chromosome 1. This is just some summary information, but we can go ahead and hit go here to actually pull up our data in the browser. And so when we press this, it will look something like this, where this at first glance this doesn't look particularly exciting, and that's basically because it's taken us to a kind of random place anywhere on the genome. It looks like the very start or very early on chromosome 1. And so what do we do once we have this? So at this point, our data has been successfully loaded in, and we can begin to go to particular sites that we're interested in. Genes that you may have come across in previous studies of your own or in published papers, and see how your data sort of stacks up to observed expectations there. And so how you do that is there's a handy search bar up at the top that lets you type in not only just the name of a gene that you're interested in, but also particular chromosomal regions as denoted by, chromosome, base pair start, base pair end, or other things. And so they have a lot of handy examples within that you can select if you so choose. But today we're just going to be using a nice housekeeping gene for this ATAC-seq data set called GAPDH, which should be in basically almost all cells very open and accessible. And so I typed in GAPDH and press go, and it takes me to this view. And so on the bottom here, we can see that these are the GAPDH different annotations, and still we're not really seeing a lot of our specific data at this point yet. So quickly to orient ourselves, here we have this is that merged peak region track that I mentioned that takes into account information from all the samples, and green corresponds to was there a peak called in that range or not. And then we have these pairs again for each sample, and so we have the stack up information or the signal information for sample 1, and then was there a peak there or not? Sample 2, peak there or not, as denoted by these orange bars. And so I want to zoom out a little bit. I want to see, I think this is a little close in for our data. And so to get a a broader sort of view, what we do is there's these handy zoom in and out buttons. And so here you can use interact with these to sort of manipulate your view and put things into a more sort of digestible viewpoint. And so I'm going to go ahead and zoom out, and now I can see the whole length of the GAPDH gene and the signal and positional information of the peaks that we've called for this gene across our samples. And so we can see clearly that there's a very consistent pattern across all 4 of them. We have peak regions in the same positions by and large. And so we have some nice data here that is very consistent. And so this is an example of a housekeeping gene, but there's other genes that are potentially of interest to your group, not only through what you've known before, but also maybe if you've performed a differential analysis and you want to say, okay, well, we found a particular site to be significantly different in regards to chromatin accessibility from one group of samples to another. And so we've performed that here. The first 2 samples were compared against the second 2. And this is sort of an example data sheet of what active motif delivery of this differential analysis may look like. And so each row here is a peak region, and we have statistical information about the tests that were performed. But what's nice and quite useful is that we also provide links to the UCSC Genome Browser directly. So once your data is successfully loaded, like we've just described, let's say, for example, we're interested in this this loci here. It has a really high or really low negative fold change, really low p-value. And so here we're seeing that this particular region might be of biological interest when we're comparing these 2 groups. And so if I were to just go ahead and click on the UCSC link column, this text here, it will take me now directly to that gene loci in the genome browser. And so we can see why this was called differential. Just by eye here, we have 2 very clean peaks in these first 2 samples, no peaks on the second ones. And so if I were to zoom out a little bit, again, up at the bar at the top, we can see sort of a wholesale view of this ISCU gene and our signal differences associated with it. And so clearly, we have a great deal of changes in accessibility as a result of our comparison. And so this is one, I think, really nice and powerful example of if you wanted to make a figure for a grant or something like that, you can very easily right-click this and save the image as presented, and move from there and adjust as fit your needs. On the flip side of that differential analysis, we also have regions that we would expect to be down in those first 2 samples and up in the second set. And so the same sort of process would apply if you're going through this table. This is what's called the merged regions table. And in it, there's these same links. And so if I go to the other side of our differentials and see sites that might be of interest, I've selected this AGBL1 region, and I click the same link. And here we go. Again, I've skipped the zoom out step just for to be reasonable and not walk through again, but we can see the same sort of clean, clear differential between the 2. And so this, I think, is the use case that is really well suited for the genome browser. You found a thing that you expect to be different between the 2 of them, and it's numbers in the table are all well and good, and that still shows that. But if you want to very clearly communicate that there are differences of interest between samples, or even in the case you want to say something is the same, these browser track views are something that people really look for and like to see, and very clearly show what you're trying to communicate in those cases. And so, yeah, I think that brings us home. So this has just been a short sort of tutorial on how to use the UCSC Genome Browser to interact with data. Here we've used an example of our own analysis deliverable, but the process should be very similar to your own analyzed data or anything else. It allows you to compare multiple libraries and experiments. As long as they were aligned to the same genome, you can add other data to this as well, be it from collaborators or previously published data, or if you're expanding a project and have something that's been previously analyzed, you can load that in along with a new data set. You can investigate specific genes or sites of interest very easily through the search bar. And our own analysis provides an even simpler view of interacting with your own data. And so I would like to thank everybody for tuning in today, and I really appreciate your attention. Excited to have this opportunity to present these details with you. And if there's any questions at all regarding this or anything else, please visit our website at activemotif.com. Thank you very much.